Hello, welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture 7, where we will discuss the theme Metabolic Phytochemistry based approaches for studying specialized metabolism. So, in the past few classes, we started discussing about the specialized metabolites, their importance as medicinal products, origin of specialized metabolites from primary metabolites, and then next how plant cell cultures can be exploited for producing the specialized metabolites. And then we also looked into the aspects of elicitation of specialized metabolism, so that enhanced specialized metabolism can be looked upon and the desired product can be harnessed. And then finally, we talked about the different methods are normally employed for analysis of metabolites. So, today we will talk about the basic aspects, that is the metabolic phytochemistry. That means, ultimate our aim is basically to make the desired product in higher amounts. So, in order to do that, we need to know the basic aspects of the metabolism. So, today I will talk about that how such studies can be undertaken. Let us go to the next slide. So, here the concepts that will be covered is about the discovery of new enzyme activities and genes of specialized metabolism. So, here I will put the maximum attention in this class, and then we will also briefly talk about the application of transcription factors which are involved in specialized metabolism, and then the following points should come under metabolic phytochemistry, that is one can go for down regulation and over expression of plant genes and its impact on natural product metabolism. Actually, this is the main target for this whole course. So, in the next class when we talk about the implications of metabolic engineering and the approaches. So, there we will talk more about it, and next point will come here is the structural elucidation of proteins involved in specialized metabolism. So, these aspects also I will not discuss in detail, because one of the previous classes I talked about it, like once if we know the proteins, if we know the structure, and then if it is possible to mutate the protein, or if during the evolution a part of the active site get lost, then that protein behaves differently, and that leads to either formation of more product or sometimes blockage of the products. So, structural elucidation of protein is very important, and then finally, the transfer of plant or microbial genes into bacterium or yeast for heterologous production. Okay. Now, what is the difference between phytochemistry and metabolic phytochemistry? Phytochemistry traditionally deals with the isolation of natural products and the structural elucidation, which involves column chromatography, HPLC, LCMS, NMR mostly, and that is the hardcore chemistry work. And metabolic phytochemistry means that say the phytochemists, they have elucidated the structure. Now, the biochemist and botanist will come here, and what they will do, they try to work out how that particular compound which was finally determined by the organic chemist or phytochemist are produced in the plant. In other words, in cell, what mechanism is operating in the cell, what are the biochemical reactions occurring in the cells, as a result of that product is produced. So, in order to know that, we need to know the new enzyme activities, and once we know the enzyme activities, then of course, one should find out the genes responsible encoding the enzyme, and then next point is that once the gene is isolated, so the scientists can play with that. So, they can either overexpress the gene, or they can block that gene expression, and as a result of that it should affect the desired natural products, particularly in terms of its quantity, accumulation quantity. Okay. And then, so these are the aspects which metabolic phytochemists shall look after, and with this brief I will just go to the next slide. So, concept of studying secondary or specialized metabolic pathways. So, this is a pathway, so A makes B, B makes C, C makes D, and ultimately P is the final product which has been determined by the phytochemists. And if the product is relatively structure is relatively simple, then the biochemist and botanist can also able to identify that using the common spectroscopic tool. So, P is the product of interest. So, it is important to know to make more P in the plant. Now, plant cell culture or root culture can make P, but what has been found, say for argument, that the amount produced there is not sufficient enough. Even with elicitation, it did not work that much level. So, therefore, it is important to look into this problem from a more fundamental aspects. That means, we need to know the enzymatic steps like E1, E2, E3, E4, these are the enzymes which convert A to B and subsequently ultimately we will get the P. So, if we can determine these activities, and first we have to see whether such enzyme activity exists or not. So, starting from the P and that is the final product and A is the precursor. So, one can theoretically predict what are the possible compounds can be formed, like P, say if it is a methylated compound, then there must be a methylation reaction occurred in the preceding step. So, that means the E4 should be a methyl transferase, and if say if it is a methylation occurred, then this step must be there must be a hydroxylation happened, because after hydroxylation methoxylation can occur. So, that means E3 should be some sort of hydroxylase. So, one can look for a hydroxylase. Similarly, if we know the precursor A and then by enzyme activity it makes B, which appears to be pretty logical. So, B to C, that may be something new enzyme activity. So, theoretically it is possible, but it has not been experimentally shown. So, it is important to really check for this enzyme activity. So, how to do that? So, one should take the cell free extract from the plant source, and then the substrates to be added, and then the cell free protein extract is the source of this crude enzyme, and if you specifically add the substrate and if required cofactor to be added, and the reaction to be carried out in the test tube, and then after certain period of incubation reaction should be stopped, and then one should check for the product formation principally by HPLC, and which needs to be confirmed by MS. And here E2 prime is basically another branching which leads to the formation of B prime. So, from B it is possible to make C, and it is also possible to make B prime. So, this must also be looked upon. So, so this one I have shown in one of the previous classes. So, Hemidesmus indicus, that is Ananthmul, so which produce the fragrance methoxybenzaldehyde, that is our target compound, and in this class we wanted to work out the enzymatic route to that. So, the plants were subjected to elicitation, and as a result of elicitation it is expected that it should produce the product in higher amount. So, this is the control, this is the standard, and this is the elicited. So, from here it is clear that elicited root extract indeed produced more of the desired product. So, that means this approach should work, and these are the different elicitor tested, and the best one was used for the biochemistry work. So, now whatever I have said just before, that if you predict, so this is the final product, the structure is known, and as you see that here it is methoxy. So, this means there must be hydroxy. So, there so this step must be a methyl transferase. So, here there is a 2 hydroxy group, here is a single hydroxy group. So, this step must be a hydroxylase. So, here it is basically a C6 C3 chain, C3 chain, and here it is C6 C1. So, there must be something reaction which shorten the chain, or that means some phenyl propionate cleavage enzyme must be there, and this is basically the structure of phenylalanine, and this is the cinnamic acid. So, here is the common or the first enzyme of the phenylpropanoate pathway, which is PAL, and which is also rate limiting to some extent. So, which should act here. So, once we more or less theoretically consider that such reactions are occurring. So, now we have to look into the literature. So, what is known? So, in the literature we got some clues that such reaction indeed exist for vanillin formation in the vanilla pod. So, now one important point what I said, that the root normally it produce the metabolite at level, but when you elicite it will produced in more amount. Now, if you add the precursors, the phenylalanine, that is the distal precursor, if that is added along with the elicitation, the level of metabolite accumulation will be far more than that with the elicitation. And now, under this elicitation plus phenylalanine addition, if you add the PAL inhibitor AOA, aminoxyacetic acid, then what will happen? So, that actually competitively bind to the PAL and will not allow PAL to function that efficiently, and as a result of that you see that the product formation is getting suppressed. So, basically this is what is showing here. So, next is the reaction which converts cinnamic acid to paracoumaric acid. Okay. So, this is what is the cinnamic acid, this is the paracoumaric acid. So, HPLC method has been devised. So, this is basically the substrate and this is basically the product. So, you see that the product formation enhanced, and no, this is basically the substrate, to substrate it getting depleted, and here you see the product formation, and with elicited treatment at different time point such enhancement of enzyme activity was noted. So, from there it is clear that indeed cinnamate 4 hydroxylase is working in that metabolic pathway, which ultimately leads to the formation of embald, that is methoxybenzaldehyde, which is the fragrant. So, the subsequently one should look for the chain cleavage enzyme, which makes the paracoumaric acid to para-hydroxy or 4-hydroxybenzaldehyde. So, which is a chain shortening enzyme. So, that is the basically the HPLC of the enzyme assay, which is showing that. And the next step is that. So, next step, this is the product of the step. So, this step for the next subsequent one, which will act as the substrate. You see, this is the product of this preceding step, and this is now the substrate of this step. So, this basically looking for the hydroxylation activity, and you see as a result of this activity it forms 2,4-dihydroxybenzaldehyde, and this is the HPLC based enzyme assay to confirm indeed that such reaction occurred at 0 reaction time and after a certain period of incubation. And the next one is that now this will become act as the substrate for this reaction, where it is the product in the preceding step. So, this now as a result of methyl transferase activity it forms this final product, and this is a methyl transferase. In order to carry out such reaction, one has to add SAM, S-adenosyl methionine, which will act as the donor for the methylation. So, like this way E1, E2, E3, E4, E5, all these 5 steps has been elucidated at the biochemical level. Now, to further confirm that, as I said that PAL inhibitors were fed, and upon feeding the PAL inhibitor it affect the embald accumulation. Similarly, the inhibitor for the cinnamate 4 hydroxylase, which is piperylenic acid, so which was also fed in a separate reaction, and that affect the embald synthesis. So, this further confirms that indeed C4H is a very much part of the component, and then HBS, then B2H, HB2H. So, for that inhibitor was available, similar experiment was done, and finally, for the last step also S-adenosyl homocysteine was added, as which is a inhibitor of OMT, and we see the suppressed formation of the product. So, with inhibitor, precursor feeding, inhibitor and the enzyme assay, So, the pathway for fragrant methoxybenzaldehyde from phenylalanine has been established at the enzymatic level. So, now what is needed actually, one should look for the genes responsible for this, so that these can be characterized at the functional level, and at later stage metabolic engineering can be attempted in the root culture system. Now, apart from this phenylalanine, the pathway actually started long ahead from primary metabolism. So, it was also important to see that whether the Sikimate pathway, which the phenylpropanoate pathway originates, whether the regulation lies there or not. So, the experiment was done that when you did elicitation, so one can check for the Sikimate dehydrogenase activity, and indeed what it was found that Sikimate dehydrogenase activity was enhanced upon elucidation, which indicate that the pathway activated at this stage only, and EPSP synthase, and there is the step here, and the glyphosate is a common very well known inhibitor of that, and when glyphosate was added in the system, and what was found that basically it suppressed the formation of embald. So, from there it was clear that Sikimate pathway modulates the accumulation. So, the regulation started even before the phenylalanine comes into the picture. So, the next interesting question we raised, in fact, we raised before than Hemidesma's work, that how 4-hydroxybenzoic acid is formed in carrot. So, 4-hydroxybenzoic acid is an important compound because it acts as a precursor for ubiquinone, for diseconin. Not only that, it is also used as a component of liquid crystal polymer, and also it is a part of the dietary phenolics. So, there is an importance to produce, there is importance, or rather there is a demand for this compound. So, it is important to know that how this is produced in the carrot. So, we raised a hairy root culture because that acts as a good model system to study, and what we have done, like similar work what I have described for Hemidesmas. So, we theoretically predicted a pathway, and then we confirm the steps using cell free extract and taking the synthetic substrates and carry out the reaction and confirm the reaction product by HPLC and wherever required by MS, and through this we elucidated the pathway. We also added the precursor, we also added the inhibitor to further confirm, and what has been shown that from phenylalanine it produces para-hydroxybenzoic acid, and from fumaric acid the one route goes to 4-fumaroyl CoA, leading to formation of different components, but in the carrot the pathway moves in this direction, not in this direction, because when you have added the MDCA which act as a inhibitor of 4-serial, it did not affect the para-hydroxybenzoic acid accumulation in the carrot. So, now let me come to entirely another different aspects where this metabolic phytochemistry can give a good answer or can solve a puzzle. You know, the tobacco produces nicotine. So, there are plenty of species Nicotiana. So, different Nicotiana species was explored for the nicotine biosynthesis, and nicotine biosynthesis basically it started with the putrescine, and then it moves in methyl putrescine, methyl pyrrolinium, and then nicotine added, nicotinic acid added, and finally it makes nicotine. So, using one species of Nicotiana or several species of Nicotiana also explored, scientists what they did actually, they fed cadaverine. So, cadaverine is basically a metabolic analog of putrescine. So, earlier what scientists they did, they fed putrescine in the culture with a name that it will make more of nicotine, but that did not happen. Okay. So, this time what they planned to use the cadaverine. Now, why cadaverine? Because cadaverine is basically the decarboxylated product of lysine, that I will come with later, but cadaverine is also a competitive inhibitor of the enzyme which converts putrescine to N-methyl putrescine, that is putrescine methyl transferase. That means, if you add cadaverine in the culture medium, which basically will stop activating the putrescine or stop catalyzing the putrescine to make N-methyl putrescine, rather it binds with the PMT, putrescinin methyl transferase. So, that is one thing. So, upon feeding of cadaverine, what scientists found that it makes anabasin in higher amount, and in the under that condition the nicotine level was suppressed. Now, why it happened? One reason is that because of cadaverine feeding it blocks here, so that the nicotinic acid whatever produced from the aspartic acid is not being utilized for nicotine, rather it channelizes in this way, and as cadaverine is there feeding. So, it makes piperidine and these two joins and make anabasin. So, that is what is called the studies with metabolic analog, and that can sometimes give new insight into the metabolic pathway. And in fact, subsequently there is another group, so where they have used which is called lysine decarboxylase, which converts lysine to cadaverine. A gene of lysine decarboxylase was expressed in tobacco, and that converts lysine to cadaverine, and ultimately it also produces anabasin. So, that means this is a very interesting work with metabolic phytochemistry, and in fact, it involves the enzymatic, the control was at the enzymatic level. Now, let us come to the discovery of the new genes of specialized metabolism and its potential metabolic engineering application. Say a bacterial gene of phenolic catabolism, a pathway characterized pathway means, okay, let me explain that pathway characterized in a microorganism which was shown to degrade some phenylpropanoid, and ultimately there will be total degradation and the product is utilized for the growth of the microorganism. So, and so that is why it is called phenolic catabolism. So, total the phenolic, ultimately the phenolic ring will be broken up. So, what has been found that this is the arbitrary pathway which produces this. Now, if you see here that this is a plant metabolic pathway where A makes B, B makes C, C makes D, and it makes specialized product. Similarly, from B there is another pathway which makes C1, and that also leads to the formation of specialized product. Now, if you look here carefully, in the bacterial system E1 is induced in this new phenolic catabolism pathway. So, E1 what is here is a bacterial enzyme. So, similar E1 is already there in the plant. Okay. So, but here it is induced, that is interesting, and second thing is this, the B which makes V. So, V may be the product of our interest, but in the bacterial system V cannot be stable, it is further getting converted and ultimately it is used up. So, if V is the product of interest, so that gives clue to the scientist that X1 is not existed in plants, but X1 utilizes B as the substrate. But if you now see the plant pathway, B is there universally present. If theoretically we transfer the X1 gene in the plant, then what is going to happen? So, X1 will compete with E2 and E2 prime. Now, it depends on the affinity of the enzyme to the substrate, it depends on the Km value, and say for the sake of consideration that if it all worked well, then it is possible that B will be channelized towards V. That means, the heterologous enzyme X1 or heterologous gene X1 will be able to utilize the substrate B and makes V. If that happens and if this enzyme is more active, then that will limit the formation of C1, ultimately this product, that also will limit the formation of these steps with this product. So, in other words, if V is produced in the plant, then these two specialized product will get affected. So, this is all theoretical consideration, but this can be tested using metabolic engineering approach, and much later I will discuss this strategy when we will talk about phenylpropanoid metabolism. I will discuss this in details. So, but this here I have discussed from the concept of metabolic phytochemistry. Now, it is also possible to block a biochemical step. So, one common way is to use the RNA interference. RNA interference mechanism I am not discussing here, we can discuss later when I will talk about the morphine alkaloid blocking. And so, what has been shown here is this, that the coffee is basically is producing lot of caffeine, and there is also a concern that if caffeine can be reduced. So, caffeine can be reduced in coffee if the caffeine pathway can be blocked, and where RNA interference technology can plays important role to achieve this. And metabolic phytochemistry is basically such basic study which needs to be done before such study can be undertaken at large scale. Next point is the transcription factors. So, transcription factors are induced as a result of activation or for signaling, which are nothing but the proteins which binds in the upstream of a coding enzyme, and then which basically influence the transcription of the coding gene. So, it is that sometimes what happens, that if you make overexpression of the genes of interest, you may not get the product of your desired level. So, one reason could be that maybe the transcription factors were ignored. So, you may not need additional copy of genes to be inserted. So, it is important if one can look into the transcription factors, and if one can overexpress the transcription factors, that may make impact on this. So, and in fact, earlier work in late 90s, people did with maize and maize cell cultures, where they have expressed several transcription factors, and they obtained very beautiful results. One such transcription factor activates, leading to formation of anthocyanin, whereas the another set of transcription factors overexpression lead to the formation of flavonoid compounds. So, that means different transcription factors play different role in activating different specialized metabolic pathways. So, this is one example, a transcription factor which is basically taken from Antirrhinum or snapdragon flowers. So, there are two, one is called DEL, which is a helix loop helix transcription factor, another is the ROS1, which is basically a MIB related transcription factor. And scientists basically what they did, they expressed these two transcription factors under control of a promoter and terminator using the same gene construct, and they expressed that in the tomato. And there are two groups working on it. One group they looked into the much later, they looked into the what happens to the vegetative part of the plant. So, that means whether the tomato leaf can accumulate anthocyanin. So, that one actually published in Plant Physiology in 2018, whereas the earlier work which is more significant as I feel, which was published in Nature Biotechnology. So, they have basically using they express this using fruit specific promoter, and ultimately the transcription factor genes are expressed well in the fruit. So, the top one, top one, this is the full fruit, this is the cut one, this is the control, that is the wild type, and this one is basically the transformed or transgenic, where the transcription factor genes like DEL and ROS1 was expressed. So, as a result of that, you see that there is a very high level of anthocyanin accumulation now in the tomato, and this is really an impact making work, and that is why it published in Nature Biotechnology. So, metabolic phytochemistry can address such important things like this, the application of transcription factors which can modulate the specialized metabolism biosynthesis. So, in conclusion, specialized metabolism is an integrated network with primary metabolism, and interfering with one reaction in this network may have unforeseen effects in the whole plant system, because there are a lot of side branching which you never know, and still the biosynthetic pathways of the most structurally known compounds are unknown. Okay. So, phytochemists they have elucidated lot of compounds, but from the biochemistry point of view, the biosynthetic pathways are not known, and this is a field of interest, and it should be occupied by the young scientists in the future, as desired by Mikey Patterson when she wrote this review in 2007. And what she also urged, that the young researchers should encourage to involve in one field, which is always interesting and worthwhile, although often hard. With this, I end this class. Thank you very much.